introduce you to Nathan. Nathan will give you a background of his interest and why he's here. But I would say that Nathan developed this assay when working through Promega. So he's going to give you a invention, a man who invented an assay that allows you to do viability. And I'm really excited by it. I haven't heard a lot of the detail behind it. And when I met Nathan, I thought this is a perfect way for me to learn and for him to explain to the rest of the industry what's going on when it comes to viability. So without further ado, Nathan.
Awesome, thank you. Can everybody hear me okay? Awesome. Well, thank you to Declan and the organizers, first of all, for inviting me to the conference. It's my first time at the Leman Swine conference. I'm a microbiologist by training, so this is new for me. But my sister is a vet and my brother is a vet pathologist, so I interact with them quite a bit. So my name is Nathan Feirer. I am a research scientist at Promega Corporation. We are a biotech company in Madison, Wisconsin. I'm excited to tell you about Viability PCR today. So just a little bit about the outline of my talk today. I'm going to talk a little bit about how pathogens are even detected, right? And a lot of you may know that. What Viability PCR is on a basic level. What's the technique? And Declan will get more into applications, specifically for viruses, for his talk. Then Viability PCR in bacteria. How does that work? That's initially what we started in inventing this reagent for, but soon after we found out it can be applied for viruses also. And then what's next? Where do we go from here with this core technology? So microbial testing methods, right? Culture-based techniques. They've been around for more than a century, going back to Louis Pasteur, right? So Petri dishes like you, sorry, that's not the laser pointer button. There we go. So things like Petri dishes, right, from Micro 101, streaking it out on a plate and seeing your bacteria show up after a day, 7 days, right? Or mammalian culture techniques for viral cultures. So advantages of this, they're relatively inexpensive, it's a low learning curve, right? You can teach an undergrad, honestly, how to do a lot of these techniques. There's regulatory acceptance, places like the EPA, the FDA, the USDA already accept these in their regulatory methods. By definition, they exclude dead microbes, right? If it doesn't, if it's dead, it's not going to grow on a plate or in a viral culture. It does provide a culture isolate for epidemiology investigations, right? So if you're trying to determine where a point source for a specific infection on a farm example came from, you can trace it back using these isolates. So some disadvantages, it can be very slow. Best case scenario, it's overnight. Worst case scenario, for some of like the mycobacteriums, it can be up to a month, right? That's a big problem to know if you have a problem, right? You have to wait a long time. There are certain bacterial states called VBNC, of viable but not culturable, where they're growing, they're metabolizing, they can make an animal or a person sick, but they're never going to grow in a culture-based system for a variety of different reasons. Big false negative problem. Complex nutritional requirements, you've heard the old adage, 99% of bacteria never grow in culture. That's pretty much true, right? Complex mammalian culture systems, if they exist at all. There are certain viruses, such as noroviruses, where culture is next to impossible. So how do you culture for it if you can't at all? So what's another method for detecting pathogens? A lot of you have heard about PCR, probably, polymerase chain reaction. And this is taking a very small amount of DNA or RNA and amplifying it to a very large amount, right? In the span of a couple hours, so you can easily detect it on something like a DNA gel right here. It's fast. It's the same day, right? You can even do this in the field, right? I mean, there are people that bring vans out to testing sites and do this, so you can really have an actionable result. It's highly specific. You can get down to the individual serogroup. This is relevant for viruses like the bird flu, right? It's flu A, but you need to know if it's specifically bird flu or not, let's say on a poultry farm. It's also very sensitive. Now, some disadvantages. Yes, you do need some more expensive equipment, sure. The biggest disadvantage for PCR, at least conventional PCR, is that there's no discrimination between live or dead or infectious or non-infectious, right? DNA or RNA doesn't really, PCR doesn't care where it came from. From a live bacteria or dead bacteria, it amplifies the same. And I kind of have that shown right here. This is some data that I took, from just our lab work, right? Where I was taking a bacteria, I had a live bacteria, I heat killed the other tube of bacteria, I did conventional PCR, right? You can't tell the difference between those two. But the one right here on the right, that's not going to make anybody sick, right? So how do we, try to get around that? How do we look at viability in a molecular sense? So that's what this slide is showing here. How do we determine it without using these culture-based methods and those disadvantages I talked about before? So kind of have to go back to the core biology question as a microbiologist is how does a virus, how does a bacteria become non-viable, right? So let's talk about bacteria first. Very common one is membrane damage, right? You burst your membrane, your DNA and your proteins burst out, you're not alive anymore, right? You're not viable. This can be caused by a lot of things like antibiotics such as penicillin or ampicillin, the beta-lactam antibiotics that affect the cell wall. Certain things like heat, right? A 95-degree heat treatment or a pasteurization heat treatment, it's going to kill bacteria. Chlorinated biocides, things like bleach or the benzalkonium compounds, those are going to affect the membrane, right? Those are all relevant conditions. So let's more talk about viruses now. How do viruses become non-infectious or non-viable per se? Is capsid rupture. That protein coat around the virus starts to break down, the nucleic acid leaks out. It can't infect a mammalian cell anymore. This is caused by a lot of different disinfectants that you'd even use in your home. UV light is a very common one, right? That causes DNA damage, right? That's why you need to wear sunscreen, right? So that DNA damage causes the DNA not to be able to be reproduced and you can't replicate anymore. And also as another one is inhibition of translation, right? The central dogma going from DNA to RNA to protein. If you disrupt that process, certain antibiotics like the aminoglycosides disrupt that process, inhibit translation, and you're no longer viable, right? So how can we use these features of how viruses become non-infectious or bacteria become non-viable to our advantage on a molecular method? So kind of going through all the methods that we were kind of thinking about, right, when we first encountered this problem. One that people have been using for a long time is flow cytometry, right? You take a small, let's say, nucleic acid binding dye that only stains dead cells, for example, something like rubidium iodide. You treat your bacterial cells with it. You put it in a flow cytometer, which is this instrument here. Basically, it's a really fancy instrument and a very expensive instrument that flows your cells past a bright laser and a camera, right? And you get two populations. That can be very effective, but it's expensive. From personal experience, the learning curve is steep, right? Not the easiest thing to do. Microscopy, you can actually put cells on slides and watch them elongate, right? They are viable if they're doing that. But that, you can imagine the throughput is very, very slow on something like that. It's one at a time, and its interpretation is very tough, right, for microscopy. Something like gene transcription can be really useful, actually, right? If you are transcribing RNA from DNA and eventually doing translation, you are viable, right? So you can measure the mRNA in cells to determine viability. That can be very effective. There are you have to be really stringent on your quality control sometimes, right? What reference gene are you using? How do you maintain the stability of the mRNA in a very standardized fashion? I'm going to talk about today, as Declan mentioned, is viability PCR. This is another molecular method. And so I'm going to kind of walk through this whole process for you. So like I mentioned before, this takes advantage of one of the main ways that bacteria or viruses become non-viable, non-infectious. The membranes of the capsids start to break down, right? And I'm showing bacteria here for representation. Here's a live cell with an intact membrane here shown in purple with its DNA genome in a mix with some dead cells, right? That gray, the membrane's starting to break down. What you can do is you can add a molecule called the Viability PCR reagent, and that's shown in red here. This has two main features. First feature is it's impermeable to live cell membranes or intact viral capsids. It can't get inside those viruses or bacteria, number one. So it's not going to get inside the cell. But what it can do is get inside these dead cells or a degraded capsid. And the other key main feature is it binds to and covalently modifies, it permanently kind of sticks to and modifies the DNA RNA from that virus or bacteria. And as a result, PCR cannot use that DNA or RNA as a template. Basically, it can't amplify that material. So what you do, in what in a well-optimized assay, hopefully what happens is you get primarily PCR signal from only intact microbes, right? So this is a very top-level view of this, and this little red dot has been a lot of work by a lot of chemists in the last 10 or 15 years. So the first molecule to do this was this molecule called PMA, or Rubidium monoazide. Some of you may have heard of it. It's not new. It came out of Montana State in 2005. or 2006, sorry. And the features that I mentioned, right? It's impermeable to intact cell membranes, intact viral capsids. And this is the main structure here. Don't worry too much about that, but it binds to DNA or RNA, and then it covalently modifies it. And it does this for this molecule by this group right up here, this N3 group. This is an azide group. Some of you have maybe have used azides as a preservative. It's a very reactive group. And specifically, once it is activated by blue light, it binds pretty much everything, including DNA or RNA, right? That prevents the PCR amplification from taking place. This is kind of the general workflow of how you use it. Pretty simple. You mix your dye with your bacterial viral sample. You have to hit it with a 30-minute blue light exposure, and there's actually companies that sell blue light boxes for this. Some people just use a light, like a UV lamp, but you can imagine that's not standardized. And then you purify your DNA, RNA, and you do PCR. Okay, so there are some challenges to this type of molecule. The main one is that blue light activation step, right? If you have a very turbid, a very opaque sample, such as a blood sample, a fecal sample, anything that light has a hard time penetrating, this reagent has a tough time for that. Or if there's a high particulate load, such as a wastewater sample, or a soil sample for that means. So why am I here? So I am in the R&D division. I'm a research scientist and a microbiologist. Like I said, we wanted to give an alternative molecule for this approach. And we call this molecule our VPCR reagent. And the general design is quite simple, right? And quite similar to PMA, where we have a domain that binds the nucleic acid, kind of gets it close to nucleic acid, so it can covalently modify the DNA or RNA. And then also, we have a selectivity group to make sure that this molecule doesn't get into intact viruses or intact bacteria. And the protocol is quite similar to PMA, but you might notice one difference, right? Do you see a blue light box here? No. There is no blue light activation needed. This cross-linking, this nucleic acid modification ability is spontaneous. And that was the main ask of, we have a chemistry group out in California, that was the main ask of them, is like, how can we do this without blue light? So I did all the biology screening, they did all the chemical design, and then after about 4 years, we came up with that molecule right there, right? And the procedure is quite simple. And we do actually have a neutralization buffer, and this is key because for any viral or bacterial detection approach, you need to lyse the bacteria or viruses that are alive, right, so you can actually detect them. You don't want this molecule to bind to live cell DNA or RNA, right? That's a big false negative problem. So you actually have to neutralize. It's only about a 15-minute step with a simple buffer so that you can detect it downstream. And no photoactivation required, right? So these are kind of the two main molecules or approaches for this type of viability PCR technique. So the principle, once again, is, remember, I showed you this before, right? Without the viability reagent, conventional PCR, live bacteria, dead bacteria amplify the same. Now, what happens when we add viability reagent to this? You eliminate the signal, right? And this is a very basic way of looking at, DNA amplification, a DNA agarose gel that you might have done in molecular biology lab back in the day, right? There's another technique called real-time PCR or qPCR. I probably guarantee almost everybody in this room has had a sample from themselves tested by this. All that COVID swab testing that we did over the last 5 years, the majority of it for the first about 3 or 4 years was done with something called real-time PCR. Basically, it's the same core technique as running something on a gel, but it's actually an instrument that reads it for every cycle of the PCR reaction. And the earlier it comes up on, or the lower the cycle number, shown on this graph here, that you get your fluorescent signals starting to rise, that means you have more bacteria or more viruses in your sample. For reference, for those COVID swabs I was talking about, anything 35 or under was considered a positive for those COVID swab tests we all did. So the lower the CT value or the cycle number means you had more of your virus or bacteria. So remember that. And what we were looking for is, okay, let's say you treat this sample, right? Your live sample is right here with your viability reagent. That's right here. A lot of amplification. Let's say you have that dead sample that you've treated with viability reagent. You get a big shift in that signal. And that's like at cycle 39, almost non-detectable, right? You can't even see that on a DNA gel. So our objective was to increase that delta CT value when I was doing all my screening for this molecule to really shift that dead cell signal to almost being undetectable. So obviously I'm not going to go through all this data, but we screened a lot of different bacteria, sample matrices, viruses, because we want this technique to be flexible, right? But as you can imagine, all these different sample matrices, from drinking water to biofilms to pus, blood, you might need some optimization, right? And you in the room and the swine producers and other agricultural producers have very different sample matrices than somebody sampling water on a swimming beach in Michigan, for example. So we've tested a lot of different viruses and bacteria, and I'll go through some of those examples now. So this is one that we actually had pretty early on from an alpha tester in Switzerland, actually in a clinical lab, where they had used PMA, another viability reagent, in blood. And PMA was working well for them in clear fluids like urine, for example. But blood was giving them trouble, so they wanted to test our reagent in that. But they wanted to look at bacterial viability in blood, right? If a patient was septic, and let's say they were being treated with an antibiotic, they wanted to know quicker if that antibiotic treatment was working. So what they did is they did spiking experiments, right? So they took Pseudomonas aeruginosa, the gram-negative pathogen, and Staphylococcus aureus that causes staph infections. It's a gram-positive bacteria. They added it to blood, they did the treatment, and then they did real-time PCR. And what they found is, one, they were able to detect the bacteria in blood using the PCR reagents. That was the most important. But an interesting result they found is that the longer they let the bacteria sit in blood, the viability of that bacteria decreased. And they're able to differentiate that. And they did other antibiotic treatments and stuff that I haven't showed. But those results were quite promising for more of a clinical setting, right? But maybe you could transition this to animal blood, pig blood. Why not? Another one that is quite common for viability testing is waterborne pathogens, things like Legionella. Right? There was another outbreak like a month or two ago in New York City. So Legionella pneumophila, it's a gram-negative bacteria, causes Legionnaire's disease. It gets in your lungs, causes pretty severe pneumonia. And it lives in things like cooling towers, hot tubs, spas, fountains. The big problem for culture-based approaches is it takes at least 7 days on a plate to grow to know your answer, right? So let's say you get a positive result, you treat your cooling tower, you're not going to know for another 7 days. So that's 14 days where your hotel may be shut down, right? So that's why we wanted to test this. So what we did with a collaborator in Canada, and you'll understand the collaborative theme is starting to show through here, well, we wanted to compare our technique for Legionella viability PCR detection versus two other culture techniques, plate culture and then IDEX, who you're all familiar with from the vet side. They have water-based testing techniques. They still have a culture-based method also. So we did spiking experiments and then looked at how much Legionella we saw. So Legio Alert, the IDEX method, was about 9,000. Plate culture was about 2,200. Okay. Now conventional PCR without viability reagent, that was actually almost up at 20,000. We were overshooting, right? That's that false positive issue, most likely due to dead Legionella. Legionella, even though it can make people sick, sometimes you look at it wrong and it will die, right? And there's DNA left over. But when we treated a sample with the viability PCR reagent that I helped develop, the value was about 4,200, right? So right in the middle of those two culture-based results. And this is why we need to keep developing these collaborations. And the main thing people always ask is, okay, it's faster, you can do it in the same day, but is it comparable to what I've been doing for the last 50 years, right? And that's where we need help to help develop this technique. So moving on to viruses, right? Can we do this in viruses? We started with bacteria, but then, well, 2020 rolled around, we all know what happened, viruses were on the brain. We were thinking, how can we apply this to viruses? Same concept though, right? Where if you have a mixture of viruses with intact capsids or viruses with ruptured capsids, that reagent is excluded from intact virus capsids. And we showed that proof of concept very early on, which that was one of the best results I ever got. And then if a ruptured capsids, it would go into that virus, bind the DNA or RNA, prevent it from being amplified by PCR, right? And for viruses, culture independence is honestly maybe more important because these culture systems are not simple. So we had an alpha tester in Spain at Agronomy Institute in Spain that did a lot of work with both norovirus, which they can culture it, but there's this very complicated intestinal enteroid system that they do that's expensive, and there's only like one tech in the lab that can do it, so we want to move away from that, right? And so they had norovirus samples, and they also had SARS-CoV-2 samples, right? And what they were doing is they were splitting those samples into two, leaving one intact, and then they were heat treating the other sample, right? And they wanted to see if the viability reagent could discriminate the difference between those two. The same general method that I was talking about before for the viability reagent. So I'll just kind of give a top-level overview of this data, but this is the norovirus data up here on the right. They actually published last year on this if you want to read more about it. But the red bars are infectious virus, and the gray bars are a virus that's thermally inactivated. It's not going to make anybody sick anymore, right? Now, untreated, without any viability reagent, those bars are the same height. You can't tell the difference with conventional PCR. Now, look what happens when you add viability reagent. Initially, we were calling it a cross-linker. It's now known as the viability reagent. Look at the difference between the gray bars and the red bars when we add a viability reagent. There's a clear discrimination. It's over like 1,000% reduction of signal of those heat-inactivated virus amplification when you treat with a viability reagent. So that was a nice result that they published. They did work on strawberry puree and other food matrices that they were working on, shellfish. Actually, norovirus can be a big problem in. They also did this with SARS-CoV-2 because at that time, if you wanted to get your grant funded, you put SARS-CoV-2 in it and it will get funded, right? So we took advantage of that, right? They had a lab in France, actually, they had the BSL-3. Once again, a collaboration. We only have a BSL-2 at Promega. We couldn't grow up SARS-CoV-2 to do this type of work. But the same thing, they did infectious in blue and heat inactivated here in green. And this is CQ value now, so the higher the bar, the less signal. Remember how I was kind of talking about that before? So without any viability reagent treatment, you can't really tell the difference, right? But when you treat the viability reagent, the difference between the blue and the green bars is quite pronounced. So we're getting a really nice discrimination or differentiation between intact and non-intact viruses. And that's where people like Declan and these collaborations that we're trying to expand on that work is big, because we don't have access to every sample type or every sample matrix or every bacteria or virus, right? So more for this audience too is how can we apply this to agriculture and vet applications, right? We've had a lot of clinical collaborations, we've had environmental testing collaborations, but what about vet med? What about ag, especially in the realm of One Health, which has become quite prominent in the last few years? And Declan will expand a lot more on this, right? But there are obviously very, very relevant viral pathogens of swine, right? Both RNA viruses like foot and mouth, swine influenza, or these DNA viruses like African swine fever. I didn't show any DNA virus work on the slides, but we've shown nice proof of concept with DNA viruses too. So, but once again, optimization and collaboration, having people with access to the sample types and the viruses, especially in a non-biased manner, right? If we can get sample types from different farms or different animal matrices that we can test that are blinded, we're not biased to it. We're just giving the data. And then another one too, and Declan will expand on this also, as you all probably know, vaccine manufacturers are moving to nucleic acid technologies, right? And this became most prominent with mRNA LNP vaccines for COVID, but even before that, right? Like some of the swine flu and the PDV, the RNA particle technology, the things from like Merck Animal Health and Sequivity, those are based off of nucleic acid technology, right? Now, could we possibly use our reagent to help do the QC and the lot release of these type of reagents? That's still one of the big problems, is they're really easy to make up in large batches, but how do you test and do your workflow development, your process development? And then also, LNPs, like I said, the COVID vaccine, and you can correct me if I'm wrong in the question and answer, I don't think there are any LNP containing like mRNA vaccines currently approved?
Don't think so.
Okay, yeah, but people are working on them, right? There are reports out of China and places in Europe where they are putting DNA and RNA in these lipid nanoparticles and showing effectiveness in animals. Now, with the current political atmosphere, who knows where that's going to be in the next 3 years, but we're trying to plan further ahead than that, right? R&D efforts will still happen. And can the VPCR help with that? So just a summary. So viability PCR is really good at reducing that PCR signal from non-viable, non-intact, non-infectious bacteria and viruses. It can work in both bacteria and viruses. And once again, I'm sounding like a broken record, that research collaboration and partnership with people who actually work with these pathogens and these sample matrices every day will be really, really useful. So thank you for listening. I'll take any questions. Yeah.
Thank you very much for the presentation. I have a question regarding to the primers.
Sure.
I think that with the PMAX, we have an issue with the length of the primer for the amplicon. Do we have that same issue here, so we can use our amplicons like a 90 BP, or it needs to be close to 150 or between 200?
Yeah, so it's a really good question. So what he's referencing is all these molecules, whether it's PMA, a viability reagent, they bind to certain targets on the DNA, right? In a typical PCR, qPCR reaction, the amount of the length of DNA that you usually amplify is traditionally between about 75 and 200 base pairs. Now, for our molecule we've tested, the effectiveness of the technique does increase as the amplicon length gets longer, but it's a double-edged sword, because the longer the amplicon length goes, you decrease your sensitivity. So we normally suggest for our workflow around 200 to 500 base pairs. And you might have to do a little optimization of your PCR assay and your master mixes and things like that, but it's definitely doable. But you make a good point. That's one optimization point that sometimes we can't control, right? So we have to be flexible, because there's regulatory acceptance of certain primer sets or ISO methods for certain primer sets that people have to, stay to. And we understand that.
Have you applied that to any intracellular bacteria, that, just one more layer of not being exposed to the treatment?
I have not. I've wanted to, right? There was a chlamydia lab down the hall in grad school that this would be really interesting for, or things like Shigella or Listeria have intracellular parts of their life cycle. I have not. It's an interesting question though, right? Because, well, then you have two membranes to get past, right? What if the mammalian cell is still active or still intact? Now, some of that comes into, and I've thought a lot about this, of like a differential lysis scheme, where you could target some mammalian component of the cell membrane that isn't present in bacteria, something like cholesterol, for example. Break apart the mammalian cells and maintain the integrity of a bacterial cell, but it's tough, right? But we've definitely been thinking about differential lysis for intracellular pathogens. That's a good point too. A mechanical lysis definitely could work. So my career started in gram-negative bacteria, and then I moved to yeast. So eukaryotic pathogen, that was a lot bigger. And the way we treated those samples had to be different, right? We couldn't use certain sizes of glass beads and things like that. But I think there would, and maybe a mechanical method would be more standardized, which would be kind of nice, right? Yeah.
Sorry, this will be our last one.
Okay. One of your slides indicates that the contact time between the Promega chemical and the sample is 60 minutes.
Yes.
Does that need to be optimized depending on the type of sample, the organism that we're targeting? And then do we also need to optimize the concentration?
Yeah, no, you make a good question about, I mentioned optimization a lot, right? Time can be a component. I think it's a lesser component. I think, honestly, the reaction happens pretty fast, my gut feeling. The bigger optimization points are, like you said, the concentration, right? If you have a dirty matrix with a large amount of bioburden, like a fecal sample or a wastewater sample, it's just going to take more, because that matrix acts like a sponge. So concentration, but also, maybe amplicon size of what target you're, looking at on your RNA or DNA genome. But concentration is very flexible, right? I mean, you can go from 1 micromolar all the way up to 100, depending on what virus or bacteria you're looking at. Thank you.
Thank you.